Bismillahirrahmanirrahim. Good evening. Today I will discuss about the nucleic acids, oligonucleotides, chemical synthesis of oligonucleotides, nucleic acid hydrolysis, and isolation and separation of nucleic acid. Before this one, we have already discussed about the structure of nucleotides and then the different types of nucleic acid like deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. So here we will discuss about the first one, oligonucleotides, and then the chemical synthesis of oligonucleotide, and then the isolation and separation of nucleic acid, which is very important. You all know about the DNA separation. So the first one, oligonucleotide introduction. In this slide, we will discuss about the general introduction of oligonucleotides. You know that the Greek words oligo mean few or small. Oligonucleotides, these are the polynucleotides whose molecule contain relatively small number of nucleotides. Oligonucleotides at short nucleic acid chain, usually consisting of approximately 25 nucleotides. And oligonucleotides are the short nucleic acid polymers used in research, genetic testing, and forensic analysis. And these oligonucleotides strategy designed to treat disease by altering the gene expression of an affected individual. You should remember that what are oligonucleotides? These are just a small chain, like when around 25 small nucleotides combined together, they will form an oligonucleotide. The oligonucleotide will contain maximum 25 nucleotide and minimum it may be 20, it may be 10 or 15 or 12 or so on. So the length of the oligonucleotide is usually denoted by mer, or Greek word which means part. For example, we have 2 nucleotide, it will be the dimer. If we have 6 nucleotide, it will be a hexamer. While when we have 25 nucleotide, then you can call it 25 mer. So oligonucleotide is just a single strand structure which having very few number of nucleotide. The number of nucleotide will be less than 25, and these nucleotide will be combined with each other by the phosphodiester bond, which we have previously discussed about it in our previous lecture. So when around 25 or around 20 nucleotide combined by phosphodiester bond and they form just a straight structure, just single strand structure, this is called oligonucleotide. And this is very important for studying a genetic material, for studying the whole DNA. Chemical synthesis of oligonucleotide. Oligonucleotides are commonly prepared in laboratory by solid phase chemical synthesis method. So in the early 1950s, Alexander Tudor's group, they pioneered the H-phosphonate and the phosphonate method, so for the oligonucleotide synthesis. And you know that this method is very important. Oligonucleotide synthesis, or this is also called gene synthesis, because gene are also oligonucleotide, short chain nucleotide, and that is used for several applications in medicine and life science research. The primers of DNA sequencing and amplifications like the polymerization chain reaction, which we also call PCR. And this is also used for the synthesis of artificial genes, and this is used as a tool for the targeted introductions of genetic mutations. Chemical synthesis of oligonucleotides is one of the most important, and you should all know about how we can prepare a particular oligonucleotide chain. This is the phosphoamidite approach to the oligonucleotide synthesis. Look toward the structure. This is very, very important. This typically simply means that how one nucleotide combined with another nucleotide form a dinucleotide, and then dinucleotide combined with another nucleotide to form trinucleotide and to form a polynucleotide chain. So this is very important. You know about the bus structure, like we have a simple nucleotide. This is attached to a solid support, just like polystyrene, which is represented by the orange. Then, like you can see this color, the round color. The phosphate group is blocked by this blue color. The nitrogenous base is blocked with this green color, and the central sugar is represented by blue line. So you can see this one. First, we will protect the sugars to stop the unwanted reactions, and then we have that sugar. You can see the OH group is unprotected. This is attached to the solid support, and then when we add with the activated monomer of another sugar, so another nucleotide, the two nucleotides combine together, and there will be a bond between that which group at carbon number 3, and then the phosphate group of that another nucleotide, and then this is the phosphodiester bond you can see here. So simply you should learn how the two nucleotides combine to form a dinucleotide, and then dinucleotide combine to form a polynucleotide. This bond is a phosphodiester bond. To prevent the side reactions, the amino groups of nitrogenous base like the dimethoxytrithyl methyl groups are diisopropylamine groups. These are used. These are called like the additional groups which are used to prevent the sugar from unwanted reaction. And this chemically protecting nucleotide is called phosphoramidite. So that's why this reaction is called phosphoramidite synthesis. And this method is called the phosphoramidite method. So in this method, we simply combine the two nucleotides to form a dinucleotide, and then trinucleotide, and then polynucleotide chain. The next one, nucleic acid hydrolysis. This is another topic. Hydrolysis, as you know, that the breaking of a bond with the addition of water molecule. You know about condensation, and you know about hydrolysis. So I don't want to discuss it in very high detail. The site of cleavage where the bond break. So in this direction, we will discuss how the dinucleotide break down to mononucleotide, simply nucleosides. So the sugar phosphate backbone are in the base. Result, what will happen? Purine and pyrimidine bases, oligonucleotide, nucleoside, ribose or deoxyribose, and phosphate group will form. Because you know that when a nucleotide, when a polynucleotide chain breaks down, so there are different possibility that either the phosphate bond will break down, the phosphodiester bond may be break down, and the nitrogen and sugar bond can be broken down to form some variety of product. RNA is hydrolyzed repeatedly under alkaline condition, but DNA is not, because RNA, you know that it contains the ribose sugar having hydroxyl group at carbon 2, but DNA don't have that OH group which cannot participate in chemical reaction. So under alkaline hydrolysis, only the RNA will hydrolyze to give different product. So if you look toward this compound, we have phosphodiester bond. We during alkaline hydrolysis, we have alkaline water. Like there will be a lot of hydroxyl ion, and then this is the chemical reaction. You can see that this OH group attack on that hydrogen of that hydroxyl group at carbon number 2 of that ribose sugar. And what will happen then? That bond between the two nucleotide, the phosphodiester bond, will once again broken down, and there will be two types of product. Look, so product number 1, this is intermediate 2,3 cyclic monophosphate derivative, and another one is short-term RNA. And with the addition of water to this compound, we can get mixture of 2 and 3 monophosphate derivatives. Sometime it will be the 2 phosphate, or it may be the 3 phosphate. So this is the central intermediate compound. This one is the basket hydrolysis that occur in RNA, and hydrolysis the hydroxyl ion SS and nucleophilic attack at the 2 hydroxyl group on the phosphorus. And what will happen? And you intermediate cyclic 2,3 phosphonucleosides which hydrolyze to nucleoside 2 phosphate and nucleoside 3 phosphate. You can see this is the same chemical reaction which we have discussed in the previous reaction. So this is the same mechanism, like the OH group will come and attack here. We will get this intermediate cyclic to 3 phosphonucleoside, and it will then, with the addition of water, it will broken down to form 2 product. The phosphate group can be attached to carbon 3. This is the nucleoside 3 phosphate, and the phosphate group can be attached into the carbon number 2, so we can get nucleoside 2 phosphate. This is nucleoside because you can see the carbon number 5. There is no phosphate group at carbon number 5. Even it have the phosphate group, but it is not at carbon number 5. So remember that if the phosphate group lie at carbon number 5, then it will be nucleotide. Otherwise, if we don't have any phosphate group at carbon number 5, then we can call it nucleoside, not a nucleotide. So this is the nucleoside 3 phosphate and nucleoside 2 phosphate. Now another one, the acid catalyzed reaction. So acid catalyzed hydrolysis, what will happen? Then the hydrolysis of glycosidic bond only at pH 3, which pH greater than 3, and the complete hydrolysis or the cleavage will be take place at pH less than 2. So when we have low pH, it means more acidic condition, and at low pH this reaction will take place. And a very extremely low pH like pH 1, it will digest the whole DNA, and that's why the stomach have the pH low to digest all the nucleic acid from the food material. So I'll look toward this compound. This is just like a simple nucleotide, and the acid produces a lot of H positive ion. And we in nitrogenous bases we have nitrogen having a lone pair of electron. It will attack on the hydrogen atom, and what will happen then? This base will be removed from the nucleotide. You can see the base S1 underneath, and we have product number B. This one just like a DNA fragment, and the product number C we have that compound, and the step number 4 what will happen, it will change into the final product like this. So in acid hydrolysis reaction what will happen, mostly the nitrogenous base will be eliminated from a nucleotide. Isolation and purification of nucleic acid, this is very important, like how we can isolate a DNA and how can we purify, because when we take a cells contain a lot of cell organelles that contain different other proteins and so many other things. So this is now the challenging task to separate only the nucleic acid like DNA and RNA, how we can separate from the cell. So to release the nucleic acid from the cell per using other procedure, must be free from contamination with protein, carbohydrate, lipids, and other nucleic acid, because we have these all these things in the cell. This process is used pure nucleic acid for testing. So we need a pure nucleic acid to remove it from the carbohydrate, protein, and other debris in the cell. Routinely isolated from human, fungal, bacterial, and viral source, we can isolate DNA and RNA from humans, from fungal, and from bacterial and virus, different viruses. And what will happen? Pre-treat to make nucleated cell available, whole blood tissue sample. We cannot obtain the DNA from the blood sample, from tissue sample, or from the blood spots. We can obtain this DNA or RNA from microorganism, and it needs sufficient sample per adequate yield. We need like sample preparation to get DNA and purify them. So this is very interesting and challenging task to remove to isolate DNA from the cell prep, from the blood sample, or from the animal, from the microorganism. So in the coming slides, we will discuss this in very high detail. The first isolation of DNA was done in 1869 by Frederick Mischer, and method used to isolate DNA are dependent on the source, age, and size of the sample. In general, they aim to separate DNA present in the nucleus of the cell from the other cellular components. Isolation of DNA is needed for genetic analysis, which is used for scientific, medical, or forensic purpose. Forensic science is very important. Like we can determine, in forensic science, we can determine different things like the heredity of a person. We can determine by the forensic analysis. We can see that the murder and the thieves and different other things. So this is very very important. In forensic science, that need to recover DNA for identification of individual. For example, there are certain individuals like rapists. There are petty thieves, the accident, or during the war victim. So we can determine from the forensic analysis the DNA sample we analyze, and then we match that DNA with the individual using database, and then we can find that this person having exactly the same DNA. Similarly, we can determine the paternity. For example, we can determine the parents of a particular kid, and the plant or animal identification. We can also identify plants and animal by their genetic makeup. So in forensic analysis, we can determine DNA by using this technique. So it means that it is very important to analyze DNA and to isolate the pre-DNA from the cell. The sources of DNA, we can obtain DNA from different sources like blood, yeast, plant, animal, bacteria. The sources for DNA isolation are very diverse. Basically, it can be isolated from any living or dead organism. Common source for DNA isolation include whole blood, hair samples, sperm, bone, nail, tissue, blood strain means blood spots, saliva, or the chick swab, epithelial cell, urine, paper card. From the paper card, we have different spots on the paper card, or a person put his hand. There is also some pigment used for sample collection, bacteria, animal tissue, or plants. So these are the sources from where we can isolate DNA and powder analysis. A store sample can come from the archive tissue sample, Frozen blood or tissue are the exhumed bone or tissue and inside human, animal, or plant samples. So it is also used in the archaeology, like we check the old people or we find some bones in the soil and we carry a DNA analysis and we can find that this particular person belonged to that place. Procedure of DNA isolation. So that is analytical chemistry. In analytical chemistry, you analyze a sample and you check, you measure the sample, how you can isolate. So what are the procedure which used for the DNA isolation? Isolation of DNA basically consists of four major steps. Preparation of cell extract. First we will take extract of the cell. Purification of DNA from the cell extract, how we can purify the DNA. Concentration of DNA sample, that how the we remove the other component and only we have a lot of DNA. Measurement of purity of the DNA concentration. So we will discuss here the first step, preparation of cell extract. To extract DNA from a tissue or cell of interest, so the cell have to be separated and the cell membrane have to be disrupted. So first we have to remove the cell membrane, and then the extraction buffer helps in carrying out this process. The cell membrane will be removed or disrupted by using this buffer. So what are these buffers? These chemicals are EDTA, ethylene diamine tetraacetate. You know this is a very well-known chemical which remove magnesium ions that are essential for preserving the overall structure of the cell membrane. And SDS, sodium lutecyl sulfate, is also used, which aids in disrupting the cell membrane by removing the lipids of the cell membrane are included in the extraction buffer. So remember that these two chemicals are used in the extraction buffer, EDTA and SDS, ethylene diamine tetraacetate and sodium lutecyl sulfate. DNA solution, this is a sample. For example, you can see this is a bacterial cell. So first step is cell lysis. Lysis means cell splitting. What will happen? This cell will be put in that EDTA and the sodium lutecyl sulfate solution. So what will happen? The cell wall will be disrupted and we will get the cell extract. Cell extract mean it contains cell wall fraction, it contains nucleus, it contains nuclear material and so many other things. Remember that cell lysis or cell splitting can be carried out by two way, either by physical or chemical mean. By physical mean, like vortexing. Vortexing mean that we will put the cell sample and some tubes, and then it put an mechanical machine which rotate with very high speed. This machine is called vortex. Sonication. Sonicator is another machine in which we put the sample and it vibrate and produce some specific sound. So this is called sonication. Chemical treatment means using some enzyme like lysozyme or using some chemical like ethylene diamine tetraacetate or sodium lutecyl sulfate. So remember that lysis can be carried out using two way, either physical or chemical means. Look to the second step. Second step, we will use the centrifuge to remove cell debris. So we will have that cell extract. We will put the cell extract in a tube, and then after centrifugation, we will get a DNA and RNA sample. Yeah, that's really important. Remember that we have another process, phenol chloroform extraction. So after getting that DNA samples, we will put some phenol and a chloroform, and what will happen? This is called deproteination. We will remove the proteins from the DNA. So phenol chloroform extraction, what will do by them? Phenol denature protein and chloroform facilitate the partitioning of aqueous and organic material. Similarly, we can use some enzyme like proteases. These degrade the protein and it can be easily removed by phenol. And using the fourth step is ethanol extraction. So we can also use ethanol to remove the protein in this thing. And after filtration, we will get the DNA material and on the paper, and the other thing will come down from the filter paper. So next slide, purification. Now this is the challenging step, how we can purify the DNA. After lysing the cell, the final step in the preparation of cell extract is removal of insoluble cell debris. In addition to DNA, the cell extract will contain significant quantity of protein salts, and reagents during cell lysis and RNA. Cell debris and particularly digestive organelles, it can be removed by centrifugation. You know centrifugation, that the sample, the mixture can be separated by on the basis of their different molecular weight. So the heavy mixture will come down and the light mixture will come to the top, leaving the cell extract as reasonably clear supernatant. A variety of procedures can be used to remove these contaminants, leaving the DNA and pure form. Remember that supernatant is the liquid portion which come on the top of the tube. Residue, well, the heavy portion which come to downside of the tube or to the bottom of the tube. The most commonly used procedure is ethanol precipitation, phenol chloropalm extraction, and chromatographic column purification, which we have discussed on the previous slide. Okay, here in this slide, we will focus on the DNA purifications. So how we can purify the DNA. The first one, culture, we will get pictorial culture, and we'll grow the cell here. After growing the cell, we'll carry out centrifugation, and what will happen, the cells, the DNA will remove the protein from protein and other part. This is the cell extract DNA concentrations. Remember that we have removed this DNA. These parts indicate the DNA, and the DNA is purified from the cell extract. This is the paper DNA. The DNA purification is ethanol precipitation. We put the ethanol in ice cold ethanol or isopropanol. DNA is insoluble in these alcohols, so it will aggregate together to form precipitate, and then it will be filtered and DNA will be removed. So ethanol precipitation is very easy. We just put some ethanol or 2-propanol and then shake the solution and then filter the solution. So the other materials will be soluble in methanol and DNA is insoluble, so it will get precipitate and it will put on the surface of the filter paper. DNA extraction with phenol chloropalm. So phenol, we have previously discussed about this one in a previous slide, like phenol denature protein at precipitate interface between aqueous and organic layer. Chloropalm increase the density of organic layer, and isoamyl alcohol also prevent the foaming. So after centrifugation of the sample, the denatured protein at stay in the organic phase, while aqueous phase containing nucleic acid. Nucleic acid is much polar, and so it is mixed with the chloropalm that removes phenol residue from the solution. So we can remove the DNA from the other proteins and other samples. Or DNA purification by column chromatography. Remember that chromatography is a very strong technique by which we can purify the DNA. So we have this glass column, we put silica particle in it, and then we will put the cell extract. Here inside the column you have silica particles, and the DNA will interact with the silica sphere. You can see here on the left side that there is round silica particle, and the DNA, these are the DNA polymer attached with these silica particle, while the proteins and these things don't interact with the silica particle, and it will go down. So the denatured biochemical protein or other compound will go down to the beaker, and then what will happen when we put water on the column, then the DNA will come out and we can separate them. This is the process of separation, which is very important. Concentration of DNA samples. After separation, we want to remove other material in water, ethanol or something, and we get just precipitated DNA. So we'll put more ethanol, and the DNA will be precipitated, and so it will be collected by centrifugation. Measurement of purity. How we can measure that DNA is pure or not? DNA concentration can be accurately measured by UV absorbing spectrometry. The amount of UV radiation absorbed by solution of DNA is directly proportional to the amount of DNA sample. Usually, absorbance is measured at 260 nanometer. At this wavelength, the absorbance of 1 corresponding to 50 microgram of double-stranded DNA per ml. With the pure sample DNA, the ratio of absorbance is 260 to 280. So it is 1:8. Ratio of less than 1:8 indicate the preparation as contaminated and there is protein or some compound. Thank you so much. Any question? If you have any question in this presentation, you can ask me through your email, or you can use WhatsApp group. Thank you so much and best of luck.